Okay, everyone. Thanks very much for coming to this week's research seminar at the Museo. This week, I'm very pleased to have with us Gabriela Montejo-Kovacevich, who is Assistant Professor in the Evolutionary Biology Center at the University of Uppsala in Sweden. So Gabriela's long-term goal is to research the mechanisms and traits that enable rapid adaptation and the consequences for population viability in the wild. And to do this, she combines genomics, ecology, and natural history. So three very interesting things for many of us there. So Gabriela's from Madrid and used to come and visit the museum here as a child, I think. But she decided to study for her biology degree at the University of Sheffield in the UK. And then she also did her master's in ecology and conservation at Sheffield. And during the period of her studies in Sheffield, she spent several summers working in the insect collection at the Natural History Museum in London. She then did her PhD in Cambridge on the ecology and genomics of thermal adaptation over an elevational gradient in Heliconius butterflies. And then she worked as a research fellow still at the University of Cambridge. This year, she has taken up her position at the University of Uppsala in Sweden. And so I'm really looking forward to finding out about some of the things that she's been doing. Okay, thank you.
Is this on now? Yeah, great. Thank you so much, Rob, for the great introduction and summary of my talk, actually. Maybe you should give it. But yeah, it's such an honor to be here. I spent my childhood coming to this museum, and actually I lived just across the road in San Juan de la Cruz. So yeah, real honor, and I hope that you enjoy my talk today. So I will be talking about local adaptation in changing environments, and a lot of the work was done during my PhD in Cambridge, and the latter part of the talk will be on my new work in Uppsala. So we live in a highly disturbed world where only 1/3 of remaining forests are pristine and where cows make up a higher biomass than all humans put together. So a key question is, how is biodiversity responding to this fast-paced change? And there's three main options. Organisms can move to greener pastures, they can stay and adapt locally to their new disturbed environments, or they can die, so just go extinct. And of course, we don't want the latter to happen, and often there's nowhere better to move or is just out of reach. So what we hope is that biodiversity can adapt locally and trigger changes that enable them to survive. In other words, we hope that they can evolve to better suit their environment. And you might have seen these satellite images before. This is from Rondônia in Brazil. And this is the Amazon rainforest been converted to cattle pasture there on the right. And this is one of those examples where adapting locally is probably going to be really hard because the environment is completely destroyed. Nothing can live there, really. So they'll probably have to move elsewhere. But there's probably other types of less intrusive land use changes that biodiversity may be able to adapt to, such as selective logging for timber production. And I actually was able to spend 3 months here in Rondônia for my master's fieldwork. And there, I was studying selective logging management, and in particular, whether you can log in a small area, here represented by the red dots, at a high intensity, and whether that was better for butterflies than logging at low intensity over a bigger area, which is called land sharing. And what we found is that there are winners and losers of both management strategies, so both land sharing and land sparing. And in a different study, we also found that strips of forest protected around rivers, which is actually a legal requirement for logging concessions in many countries, do successfully protect some butterflies, but not all of them. So that indicates that for some species, untouched, pristine forest really is irreplaceable. So these studies are useful at telling us the whole community responses to environmental change. But they don't tell us much about why are some species able to adapt and others just perish. So I think we need to look at local adaptation in individual species to be able to make these generalizations about winners or losers of environmental change. So just some definitions. Local adaptation occurs when there's genetic change and individuals from a population have higher fitness than in their local environment than those from other populations of the same species if they were moved to that environment. So divergent selection across a landscape can favor evolution of local adaptation because the populations are experiencing contrasting like environments or conditions. So an example of this is, for instance, the pocket mice in North America, where they have adapted to volcanic lava backgrounds with mutations that make them black. And if you were to move those individuals to the wrong background, they would be selected against, and they would have much lower fitness than in their local environment. But a lot of environmental variation in the wild is not binary, right? So the environment changes gradually along clines where biotic and abiotic factors change, and populations have to adapt to that. For example, in this classical experiment, they took seeds from yarrow plants from different elevations and then grew them in a common environment and found that the differences in height that they observed in the wild were maintained, and therefore this was probably quite a heritable and adaptive trait. So given that we can study local adaptation and that is very important in nature, how can we go on about doing that? So at its core, studying local adaptation is interdisciplinary because you have to link ecological factors and their evolutionary outcomes. So in my study and my work, I try to look at it at different levels. So in the environment, we need to define it, what's changing and by how much, and then identify the actual traits that confer adaptations, and then also look at the genomic variation across space and figure out what promotes local adaptation or what hinders it. So today I'll tell you two stories, adaptation to altitude in Heliconius butterflies, and one is in Euphordias butterflies in North America that are adapting to climate change. So the first story is on Heliconius, and it's work I did during my PhD with Chris Jiggins and Caroline Baquet in Ecuador. And you might have heard of Heliconius for a bunch of interesting traits. They're, for instance, the only butterflies that feed on pollen as adults. So that gives them an amino acid source and allows them to live really long lives, up to 9 months in the wild. They also have really large brains and really good memory, thanks to these mushroom bodies, because they follow the same line traps every day and can find specific flowers throughout their life. And of course, they have this Mullerian mimicry, whereby different species look exactly the same to protect themselves against predators and warn them that they're toxic. But what you might not know is that some species are very widespread, so they're found all across the continent and across many elevations, whereas others are restricted to either the highlands or the lowlands. Like here, Telecife is restricted to around 3,000 meters up in the Andes. And Heliconius species richness is highest along mid-elevations in the Andean mountains, and this probably hints that it facilitated ecological speciation in this genus, and it's played a major role in speciation and adaptation. And mountains are really good settings to study local adaptation, in my opinion, because the physical environment changes a lot over a very short space, and that increases selection pressures on the organisms living there. And then with warming climates, we are seeing that they're having to extend their elevational ranges upwards, and habitat degradation in the lowlands is really forcing organisms to find refuge in mountains, as is the case in Borneo, where all palm plantation in the lowlands is destroying it. So it's becoming increasingly important to understand the potential of organisms to adapt to altitude, as well as identify the traits that allow them to colonize those elevations. So I took a range of approaches to gain insights into how these butterflies adapt to altitude. So let's first talk about the environment. The ecologist in me really wanted to know, how much actual difference is there in this moderate elevational clines that some of the butterflies inhabit. And here's Mashpi, one of my sites. It's a cloud forest in the western slope of the Andes. And you can see that it's a very complex habitat, right? So you can imagine the butterflies flying in and out of this forest, looking for mates or host plants, or laying eggs. And at the beginning of my PhD, I wondered whether I could just use publicly available data sets that interpolate data. But of course, a lot of these are based on weather stations, like this one in Ecuador, and they're specially designed to minimize the impact of the forest on the readings, right? So they're always found in places where there's no forest. And that, of course, can give you quite an inaccurate picture of the temperatures within the forest where the butterflies actually live. So what we did instead was record temperature and humidity in 28 sites across both sides of the Andes. And the high sites are in blue and the low in pink here. And we did this for a full year, and we placed 64 data loggers. We had some in the understory, which you can see one there, and then we put them up in the mid-canopy at about 11 meters, which is more or less where the butterflies fly, but also it's the highest we could get them. So this paired approach allows us to directly compare temperature and humidity variation throughout the day in different parts of the forest that the butterflies experience throughout their lifetime. So one of the most physiologically relevant metrics we looked at is the range of temperatures experienced throughout the day by the butterflies, which is in the y-axis here, so it's temperature range. And what we found is that the temperature range is generally higher in the lowlands, and that the difference between the canopy and the understory is also higher in the lowlands. So that's the difference between the triangles and the circles. And you might expect this, but actually, if we had not gone through this whole effort of measuring it in the wild, we would have got quite a different picture. So these are the results from looking at WorldClim 2. And as you can see, they're very overestimated, which you might expect because it's a weather station without forest around. But not only that, the trend is the opposite, right? So it's predicting that in the highlands, they experience a higher temperature range, which is just not the case. And that's not to say that WorldClim 2 is rubbish. It's just that to study small organisms, especially in the tropics and in high elevations where there's very little data to interpolate from, you should really be looking at microclimates. And another important conclusion from this study was that climate variation across eastern and western slopes of the Andes is quite similar. And that's good news for my later studies that I'll talk about. Okay, so we can conclude that the forest buffers temperatures within, but there's still quite big climatic differences between the highlands and the lowlands that these butterflies are having to adapt to. So what are the phenotypes that allow them to succeed across such a big range? And the first obvious trait I was interested in was heat tolerance. And I measured heat tolerance in 10 wild species, some that were high or low elevation specialists, and others that were found across the whole range. And to test this, I just carried a kettle around Ecuador and a box and lots of thermometers. And the protocol is as follows. So you put a butterfly in a glass pot, and you heat the pot up to 39 degrees, and then you maintain that temperature, and you wait until the butterfly gets knocked out and loses the ability to walk or stand. And then you measure the time and take the pot out, and they recover. So that time is the knockout time that I'm going to talk about. The y-axis is the knockout time, and the higher, the more thermally tolerant a butterfly is. So what you can see here is that clearly in the lowlands, butterflies tend to be more thermally tolerant, so they can, they take longer to get knocked out by this high temperature. But what was quite shocking is that nearly half of the individuals of the 3 high-altitude specialist species actually didn't even make it to the experimental temperature of 39. So that's why there's so many zeros down there. And in our microclimate data, we found that at least once a week, temperatures went over 30 degrees in the highlands. So these species must be behaviorally adapting to avoid these dangerously hot times of the day. But of course, with climate change increasing temperature extremes, it might be a problem for these high-altitude specialists. But there's other traits that vary with elevation and that are very important. So let's take this cline that's about 800 meters in elevational difference. And the atmospheric pressure drops by 10%. That translates into thinner air, less oxygen, and there's also a big difference in forest structure as it becomes denser, more like cloud forest. So a really important trait for butterflies is flight, because it's energetically super costly, and it's under very strong selection to be optimized for the local environment. So I became interested in wing shape, and that determines flight mode, and we used samples collected for my PhD, but also that were stored in Cambridge since the 90s in envelopes, and we photographed them and then wrote some scripts to analyze them automatically and get this aspect ratio metric out of them. So that will tell you, I think the pointer's out of battery, but yeah, it will tell you how elongated the wing is by giving you the major and the minor axis. So here at the top, you have an example of an elongated wing with a high aspect ratio, and at the top, you've got a species with a rounder wing that has a lower aspect ratio. And then in the x-axis is the mean altitude of the species. And you can see that there's a negative correlation. So in the highlands, wings tend to be rounder, which is pretty cool. But then we wanted to look at the species that inhabit the whole range, right? The ones that have a long horizontal line. And for that, we took 2 species, Errata and Melpomene, and turns out that the same trend is maintained. So that in the highlands, they tend to have rounder wings even within species. And why rounder wings? Well, we haven't personally functionally tested it, but from other studies in other systems, the idea is that it could help deal with this reduced air pressure by increasing lift during flight, which is very important for it to be energetically optimal. So we find both traits, heat tolerance and wing shape, varying with altitude in the wild in the species, and that brings about the question of how plastic or heritable these traits are. So, of course, plasticity can be genetic and adaptive, but for this example, we're just going to make the distinction between traits that are fixed within a generation and that are locally adaptive, and traits that change within a generation and can be considered plastic. And in an ideal world, to test whether they increase fitness, we would perform a reciprocal transplant, where you move individuals from one population to another, and then you test their fitness in the wild. But this is just not feasible in most study systems. So what we end up having is this series of observations in the wild that show that there's a difference between populations, hopefully with several replicates and species. And then the next step is to do common garden rearing to tease apart whether they're genetic or plastic. So for that, you rear individuals in common conditions, and in a locally adaptive genetic trait, you shouldn't be a difference. So you would expect them to maintain their native trait value in common garden. Whereas with a more plastic trait, they should be converging into a mean trait value. So this is what we did. We collected females from the highlands and the lowlands, and then we brought the mothers to an insectary that we built in our collaborating university. And then we reared caterpillars in parallel and in exactly the same conditions, and then looked at the phenotypes in the offspring. So this is the results we've already seen, that for one species, Heliconius aurato, in the highlands, they have much lower thermal tolerance. And when you bring females to a common environment, their offspring actually end up having the same thermal tolerance. So there's no difference, regardless of what altitude the mothers came from, regardless of their genotype. So this indicates that thermal tolerance is probably quite a plastic trait in this species, but we can't really tell from these experiments whether it's just acclimation in the adults or developmental plasticity irreversibly determining thermal tolerance in the adult. So what about wing shape? We know that there was a difference in aspect ratios, so the highlands, they're rounder, and when we bring them to a common environment, actually the difference gets even stronger in the offspring, and this suggests that it's quite a genetic and heritable trait. So what we can say is that thermal tolerance is probably quite plastic, and that wing shape is probably quite heritable. So to link phenotypes and genotypes, we can carry out a genome-wide association study where you're just trying to find the genetic basis of a trait. And that is, these kinds of methods are good for looking at one trait at a time. And I won't talk about these results more today. And instead, I want to talk about a different approach where you say, okay, let's completely forget about the traits and instead look at everything and try and find genetic differentiation across populations in different environments and do this in a replicated manner across space and try and find the interesting candidate genes like that. So studying repeated adaptation to similar environments is a powerful approach to look at the genomics of local adaptation. And there's an obvious methodological advantage because it provides this natural replicated experiment that can help distinguish between locally adaptive loci or neutral and beneficially, global beneficially mutations. And biologically, it's also very interesting to study repeated adaptation because you can ask, does evolution take the same path over and over, or do they come with different solutions for the same challenges? And if so, what are the mechanisms that allow this parallelism to take place? And mountains, I think, can provide the perfect settings to do this because you can get populations that have repeatedly and independently adapted to similar environments. So you get replication across space. And then if you work with speciation taxa like Heliconius, you can get replication across lineages. And then you can look at whether different species are doing the same thing or not. So we did that in Heliconius by sequencing whole genomes of two widespread species, Errata and Melpomene, along 4 altitudinal clines in Colombia and Ecuador, and on both sides of the Andes. And the Andes here in red act as a barrier to gene flow. So they can't, at these latitudes, move between sites. So we hypothesize that they have independently adapted to altitude on either side. And to increase our power further to detect these divergent selections, regions potentially under selection, we took a 3-population approach. So we have a highland, a lowland, and then a low-distant population, and that helps you control for the effects of geography in the genome and drift. So this would have been impossible without lots of collaborators, so I just wanted to mention them here and thank them. So let's make some predictions about the repeatability of evolution. Generally, the more divergent lineages are, the less you expect them to have molecular parallelism, right? So they will share less standing genetic variation, and they're more likely to come up with different solutions for the same problems. So we expect that those transects connected by a gene flow, here called replicate transects, should have the highest likelihood of molecular parallelism. So repeatability, because they share a larger gene pool between them. And then as we move to allopatric transects on each side of the Andes, they should have less parallelism. And then when we look at 2 very divergent species like Heliconius, Errata, and Melpomene that diverged 12 million years ago, you expect them to have even less parallelism because they share less of the ancestral variation between them. So to identify genomic regions diverging across these clines, we use population branch statistics, which uses these 3 populations And when you make a tree for the whole genome, you can already see that the highland population has a longer branch. And this is longer than you would expect from isolation by distance alone. So it's not just geography that's making them different. There's potentially lots of local adaptation also going on in the highlands. So now I'm going to show this branch statistic for high altitude across the genome in 4 transects, but only on one of the species, Heliconius aurata. And the peaks that are shared within sides of the Andes will be colored in green or in blue, and then the red ones will be across sides of the Andes. The first thing to notice is that there's many peaks, right? So there's a lot of regions that are potentially providing adaptation to altitude, and that they're more different in the highlands than you would expect by geography alone. And the second thing to notice is that many of these peaks are in the same genomic regions across the different clines, and that's why they're colored. And most of them are colored in green or blue, so they're shared within sides of the Andes, but actually quite a few of them are also shared across sides of the Andes in red. And a similar picture was found for the other species, but there was no parallelism between the 2 divergent species at all. So that matches up with the general predictions that we made in the previous slide. But a second, perhaps more interesting question for me is we can ask, like, why is there so much parallelism? What are the mechanisms that are facilitating it in this group? And one option is that they're all independent mutations that have happened repeatedly across populations. Another option is that those mutations already existed in the ancestral populations, so they were standing genetic variation that got repeatedly selected at high altitude in different populations. And finally, a third option is that those adaptations were stolen from other already adapted species, species that were already adapted to high elevation through hybridization and adaptive introgression. And to be able to look at this last question, we sequenced 85 individuals from 5 high-altitude specialist species from the Heliconius genus. And what we found is that the majority of the molecular parallelism can be attributed to standing genetic variation, so variants that were already there, or gene flow between populations where there can be gene flow. But a large proportion, actually, we can attribute to adaptive introgression from closely related species that were already adapted to high elevation. And this realization that adaptive introgression is important to adaptation is not new, definitely not within Heliconius, because the color patterns have been readily shared between species. But there's also other examples. For instance, snowshoe hares get their brown summer coloration from jackrabbits, or introgression of melanism from dogs to wolves. But I think perhaps what's more novel here is that we're looking at a whole suite of potential adaptations to the environment rather than at a single trait, and we still see that hybridization is having a huge role. So introgression probably speeds up adaptation. Instead of having to wait around for the correct mutations to arise, you just steal genetic combinations that are already adapted to this environment. So the scans suggest that there is repeated evolution, and that standing variation and introgression has an important role. And all in all, I think our work shows that there is local adaptation to altitude in Heliconius, which hadn't really been studied before, and that you can look at it from different angles. But this is all about studying change across space, whether at the environmental or phenotypic or genotypic level. But at the beginning, I said that to understand the mechanisms driving local adaptation and to use that to predict species responses to anthropogenic change, a really important aspect is time, right? So how quickly can organisms adapt to a new or a changing environment? And unlike what Darwin thought, evolution can be fast. There are many cases where we have observed rapid evolution in the wild. For instance, COVID was evolving before our eyes. Every day on the news, they were showing new variants that were appearing across the world, and only those providing with the highest fitness for the virus, in this case, escaping vaccine immunity, were the ones that remained in the population and became prevalent. And even speciation can be rapid, right? So with recent adaptive radiations like cichlid fish, or even through hybrid speciation, where, for instance, in finches, a single hybridization event 30 years ago has given rise to a new species, this big bird lineage. And we're also getting more examples that are really cool from experimental evolution in the field, like this paper that came out last week, where they forced evolutionary change by transplanting snails to a new environment for that ecotype and follow them for 30 years. So if evolution can be fast, it's not so crazy to suggest that biodiversity might be able to adapt to anthropogenic change, right? Or at least part of biodiversity. So in the first 2 chapters of The Origin of Species, Darwin made this distinction between selection and domestication and selection on natural variation, and argued that in nature, change was very slow and gradual. I think this difference between natural selection and artificial selection is becoming increasingly blurred, because humans are altering the environment to such extent that selection is now really strong and comparable to domestication, where selection is done on purpose and super strong. And there's examples of adaptation to anthropogenic change, like, for example, evolution of insecticide resistance, or to pollution, like in Atlantic killifish, where they've adapted to previously lethal pollution levels in urban estuaries. But adaptation is not always necessarily good in the long term. For instance, in Edith's checker spot, Singer and Parmesan described the extinction of a butterfly population that had adapted to an exotic plant brought about by cattle ranching, and then when the ranchers left, the plant disappeared and the butterflies went locally extinct. So they can adapt fast, but that can lead them to these evolutionary traps that they maybe can't get out of so easily. So a key question I would like to ask in my lab is, what facilitates rapid adaptation at the genetic level, and what are the consequences for evolutionary change, so that we can really get at identifying winners and losers before it's too late. And I've been doing that by focusing on systems where there's rapid adaptation potential, studying genetic variation across time. So one of the work I've been doing during my postdoc is with invasive moths in Brazil that have hybridized with a local species and shared insecticide resistance genes between them, making them terrible pests and causing yield losses of billions of dollars, and they even declared a state of emergency in Brazil because of it. And also I've been looking at the introduction of biocontrols in the South Pacific as a natural, artificial experiment of evolution. But today I want to tell you about my newest project, which is adaptation to climate change in the Edith's checker spot. So I don't think I need to tell you that the last 10 years have been the 10 warmest years on record since the records began, and that 2023 was 1.4 degrees hotter than the pre-industrial average. And a 1.5 degree increase in temperature is predicted to put 14% of terrestrial species at high extinction risk. But these predictions don't incorporate the potential for populations to evolve, nor the idea that they could fall in an evolutionary trap. And this is partly because we know that organisms can adapt to climate variation within their range, like I showed with Heliconius, but there's many other examples of that. But there's very few examples of direct adaptation to climate change that we have observed, really very few. So I think we must study the role of evolution in preventing extinction, and for that we need a better mechanism understanding of the process of rapid adaptation. So this is why I've chosen Euphridias butterflies, and it's funded by SciLife, and it's going to be most of this work carried out in my new lab in Uppsala. And this time, I cheated a little bit because I found a system where we already know an awful lot about the phenotypes and the ecology and how they've changed through time, thanks to decades of work by Mike Singer and Camille Parmesan. And actually, in the last few years, a genome has been assembled by a group in Stockholm. So really, in collaboration with them, the stage is set to look at the genomics of adaptation. So euphridias live in North America, from sea level to about 4,000 meters up in the Sierras, and they use a range of host plants, quite a few. But here I'm just going to focus on two. There's Colinsia in blue and pedicularis in yellow. And different populations of euphridias have very strong preference for either of these plants, even though the plants are found basically everywhere. So they could use the plant everywhere, but they don't. In some populations, they prefer one, and in others, the other one. And that's what you can see here in this map. So Mike and Lindy McBride did some really neat studies showing the host of adaptive traits that enable them to exploit and prefer these different host plants. But today I'm just going to focus on one trait, and that's oviposition height. So they showed that oviposition site is adaptive. So in Colinsia, the blue one, they prefer to eat the young leaves. So the adult females lay their eggs high, because then the caterpillars will eat the top of the plant. Whereas in pedicularis, they lay low, because they're trying to avoid incidental predation from grazers, like deer in this picture. And grazing is pretty extreme. So pedicularis is a hemiparasite of pine trees, and it remains delicious and juicy even during heat waves. So it's a very important source of water and nutrition for these grazers. And any legs laid on the top, as you see in this picture, will have been just taken by the grazers incidentally. They've also shown that oviposition height is heritable. So they did this by crossing individuals from different populations that were adapted to the different host plants. And then they tested oviposition height preference in the mother hybrids. And what they showed is that it seems to be quite heritable, and that laying low is recessive. So the hybrids here in green are actually laying high, like the blue ones in Colinsia. Yeah, just a note to say that they have one generation a year. So actually doing these experiments took many years, and kudos to them for doing it. And this is a video I took this summer of a butterfly as a classic drop-air behavior. So she lands at the top of the plant and then immediately, smells it, thinks it's good, and then starts dropping down to find the lowest point in the plant where she can lay an egg. They're quite stubborn with their behaviors, which is good, but they're also quite picky, so they can spend a long time just trying to decide where to lay the egg. Okay, so where is the catch? This seems like a great adaptation to avoid predation, and why not just keep it that way? Well, the issue is that the ground is very, very hot. So this is Camille Parmesan this summer, and she's kneeling in front of a pedicularis plant. And this is during a heat wave in Rabbit Meadow, so at 2,300 meters in elevation. And you can see that the plant itself is pretty cool. That's because, it's stealing a lot of its water from the pine roots, so it can stay cool and juicy. But the ground is super hot. So if you zoom in, you can see that the plant is quite cool, at around 30 degrees, but the ground can get up to 72 degrees. And a previous experimental study showed that the eggs can survive for 1 hour at 45 degrees, which is already quite a lot, but at 48, they get completely killed. So this is, you can see, a very delicate balance, because if the eggs are laid too close to the ground, they will get burnt and just die. So in these two populations, Paiute and Rabbit Meadow is a typical behavior in the past where they would lay low, as you would expect, and that's what you see down here in the egg height. But the ground is getting hotter because of climate change. So what they've observed is that they're now laying higher. They're shifting towards laying at the top of the plant, despite the risk of getting predated by the grazers. So much so that they're now laying on average 7 degrees cooler than they used to lay when they were laying close to the ground. And that's kind of overshooting current levels of warming. So within population, using the same host plant, there seems to be a shift that's protecting them against current and future climate change. So there's this trade-off between escaping incidental predation from grazers and escaping the lethal heat from laying too close to the ground. And I heard Mike talk about this story in ESAB 2 years ago, and I thought it was fascinating, so I went this summer to see it for myself. And I spent a couple of weeks lying on the ground, looking at these butterflies lay eggs. And I did it in a population that's currently polymorphic for the trait. So some lay high and some lay low. We tested about 100 butterflies. We did it blindly on different plants, different days, and found that the behavior is highly repeatable, especially for a behavior, is about 45%. And that indicates that it's quite heritable within the population. And that, of course, made me excited about sequencing these butterflies and trying to find the genetic basis of the trait. So we have sent one plate of extractions for sequencing, and I hope to do a genome-wide association study and find out the genetic basis. And we're also looking at genetic differentiation between populations that are using different host plants and that they're adapted to laying either high or low. So my plans are to do a selection experiment in the field to look at egg survival at different heights with grazing pressure, and also to use museum collections to look at specific genetic changes in populations through time and with different environments. And it would be really interesting to look at other effects of climate change, such as reduced gene flow between populations because of all the crazy fires across California, and see whether any more evolutionary traps are in store for these species. So all in all, I think this system can provide new insights into the genomics of adaptation to climate change and link phenotype and their genomic basis and function while learning about rapid adaptation in the wild. So I hope I've shown that these are very exciting times to be looking at local adaptation. And what I have found is that ecological opportunity is, of course, very important, and that we need to understand the environment to be able to see it. And that existing genetic variation and gene flow are probably crucial for fueling rapid adaptation. And that strong selection is also needed. And of course, if you can, you should be measuring it in the field to really get a good idea of where we expect evolution to take place and how quickly it will, whether it will be quick enough, basically. And I'll be hiring soon, so if you're interested, please chat to me. And with that, I just want to thank all my amazing collaborators and the butterflies and you for listening. Thank you very much.
Thank you very much for an amazing seminar. Right, okay, everyone. So give yourselves a few moments to chat with the person sitting next to you about what kind of question you might like to ask. Yeah? Think. Anyone already? Oh, Ugo's got one already. Okay.
Hi. Thank you. It was a nice and really cool presentation and talk. So, in your common garden experiments, do you control for maternity effects, and have you thought about going on different generations to look after what is going on?
Yeah, no, that's a really good question. So maternal effects are hard to control for in this setting because we don't have the fathers, because we collect females that have already been fertilized in the wild. That's just how it happens. Heliconius, sometimes they even mate before they exit the pupa, right? The males are there like waiting. So then we don't have the father genotype. If we did it, then it would be easier to make the parent-offspring relationships and look at heritability that way. Mother-offspring relationships are really strong, and that, again, suggests that it's quite heritable, but we can't really tell maternal effects apart. Keeping them for more generations is really hard. I mean, this is in the forest, and things die and there's disease. But we did, and I wasn't really involved in it, they carried on with this experiment and did a reciprocal transplant experiment with climate chambers and have found that these results are still the case. So the thermal tolerance seems to be quite plastic, at least within this very widespread species, right? We haven't looked at those that are, very well adapted to high elevations. And wing shape is very heritable. Yeah.
Hi, thanks a lot for a super cool talk. One thing I was thinking about is, do you think that simpler genetic architectures of adaptive traits make it easier to adapt faster, or because there is also a risk that drift will impact the trait much higher, right? So is there a link between the two, or?
Yeah, I mean, genetic architecture is like the hot topic, and it's so important. I think it depends on the system. So for instance, in Euphridias, if you need 10 traits, let's say, to be well adapted to a host plant, I would suspect that a genetic architecture that keeps those variants together, such a big inversion, for instance, would be favored. And if that appeared, then that could really speed up the process. But you're right that drift, you also have to counteract the effect of drift on architecture. So I think it'll depend on the nature of the trait. And we've definitely seen examples where, a big inversion arises and stays because it has all the beneficial stuff within it, or even through introgression of inversions that can, protect them against recombination. Splitting everything apart. But it's a really interesting thing that I will definitely look in Euphridias. Yeah.
Thank you.
Hi, thanks again for your seminar. I love to see the combination of field work and lab work and genomics and all.
Thank you.
Such a pleasure to see. I have a question with the Euphridias butterflies. You showed us that the populations were different genetically, those that preferred to lay their eggs higher in the plant versus lower in the plant. When you did your experiment and were able and showed that with climate change, they were those that laid below were moving a little bit upward. Do you see other traits related to this? Because you've already shown us with the Heliconius that the leaf size or shape was important. Do they need to have something else to be able to go down there and lay their eggs? Or
we really don't know enough about this behavior, and that's something that I tried to get a sense for this summer while doing the experiments, is how to define and describe the behavior well, because it's not as clear-cut. And of course, in the end, what matters is where the egg is, right? And the height might not be a good representation of how hot it is, because in the middle of the plant, it'll be cooler than if they lay it. Yeah. So I think there's potential for evolution to go in different directions within the population that are adapting to the same plant, because, as I said, laying in the middle might be as cool as laying, at the top. So yes, we will have to dissect the behavior much more. And the other part of the story I haven't mentioned is that laying low has another advantage, which is that you develop faster because it's warmer. And developing faster is really good news when you've got 1 generation a year, because otherwise you might get snowed in and miss out on mating, or you might be really late and then there's no one around. So it has really interesting effects on population dynamics as well, if they're really shifting their height, and that will slow down development. Yeah.
So do you monitor the population size also to get an idea if they're declining? Or?
Yeah, not really. I mean, the current monitoring is Mike Singer, who has gone to these populations for decades, and he's 80 now, and he knows and he can tell. But no, we haven't had a good measurement of density. There are public records on iNaturalist that I want to use, but probably not enough to get a good idea of the population density.
Thank you.
But we'll try.
It was, thanks, nice data and nice talk. I was just following this question. It's about this model system of the position and the plant. I wonder if also the plants are very plastic, so they will also adapt to hotter soils. So they might change also their shape.
Yeah.
So you should take care of that also.
Yeah, yeah, exactly. I think, and also, as you said, with the work of Heliconius and looking at microclimates, it makes you realize how quickly temperature can change in very small spaces. And we will have to look at that for sure. I think the fact that they take their water from deep roots in the pine does help with keeping the plant cool, but it might evolve as well because it is, in the end, close to a really, really hot ground. But yes, I think from now on, when we do experiments, I'm trying to come up with a way of having thermal probes along the plant that don't disturb the plant or the eggs, because eye buttons are a little bit too big, and just leave them there throughout the whole season. Because that's the other important thing, that I think selection is happening when there's a heat wave, where temperatures just go crazy and the eggs are there and they can't move. So we need to be there for those times across many plants and look at variation.
Thank you.
Yeah, thanks.
Thank you very much for the talk. It really always warms my heart when I see somebody that has treaded the same beautiful paths in my homeland in Ecuador. So the elevational gradient of the Andes is such an amazing model. So I have 2 questions. One quick one. What was your highland location on the eastern side? Was Baeza, Yanayacu?
Yes, we had a couple, but we were like basically up from Tena.
Up from Tena.
Yeah,
okay.
yeah.
That makes sense.
So that kind of, and then our distant lowland, we had Yasuni.
Yasuni, of course.
And our close lowland, it was like, lowland from Tena, basically.
All right.
Because Tena is where we had the lab and,
Oh, yeah, okay, that's good.
Ikiam.
Okay, makes sense. The other one is, okay, so the changes in diversity and composition of the Heliconius across elevations, how much do you know how much that is caused by host plant differences in changes in host plant use? And then do you have an idea of how plastic these preferences are, and therefore how quickly they may evolve to shifts in host plants?
Yeah, that's a really important burning question. In Erato and actually in Melpomene, in these populations, the host plant often goes higher than the butterflies. So that shouldn't be a limit, at least to elevational range. But definitely how within the genus, how they use host plants and the shifts that have happened within is really important. And the truth is that we often don't know what they're eating.
Yeah, makes sense.
We know, that they prefer, from having them in the lab, they're like, oh, they prefer this one, so they might be eating this one. Mm-hmm. But actually what they're doing now is looking at the chemistry, because they sequester compounds from the plants. Mm-hmm. So they're trying to figure out, especially in Brazil, like what are they eating and how does that look? And it's definitely, for the speciation of the genus is a huge factor, because they've co-evolved and they're so specialized on passiflora that,
Thank you.
We might have time for one more question if someone... No? Okay, great. So if you do have questions, you can do them at the kiosco over a beer now.
Nice.
So thank you very much.
Thank you.
Really great seminar. And yes, we have another seminar next Friday, I think. It's a Seminario Polar, no? I think so. Yeah, so we have a polar seminar next Friday at 1, that'll be at 1 o'clock. So anyway, let's go and have a beer. Okay, bye.